We were told that the mRNA from the vaccines would only last a few hours, perhaps a few days, and that the resulting spike proteins would only last for a few days. Both of those turn out now to be proven to be spectacularly wrong. Hello everyone, Dr. Chris Martinson here. Thanks so much for coming back and being here with us today as we talk about something that's actually a pretty shocking finding, and we're going to go through the data for it right now. We've been getting more and more reports of people who've had experienced difficulties from the safe and effective vaccine that we're finding more and more out there now, that there are side effects for some people, and I think we're getting an answer to that now, and it rests, I think, at least in part, with understanding about the dynamics of what happens after the mRNA lipid nanoparticles, the LNPs, are injected into the human body. So we're finding that out more and more now, and so let's go to this one. This is episode 47 today. We're talking about mRNA and spike proteins. They can persist now up to, I dropped out an important thing here, 8 weeks. Let's put that in there. Let's pretend I meant to do that. It's a shocking finding because I surprised you with it. 8 weeks is the answer, and this comes to us courtesy of a really, really awesome paper we're going to go into, which is this one. It's in a great journal called Cell. Very, very elite. This is a peer-reviewed piece of work. Very nice. A lot of authors on there coming from really great places. But first, let's talk about something so that we all have the same context. RNA and DNA. So what are we looking at here? Let me get my drawing tool out real quick. This here would be the cell boundary that we have here, and above this red line would be the inside of the nucleus, and out here is the inside of the cell called cytoplasm, and out here would be the rest of the world compared to the cell. So inside the nucleus, that's where the DNA lives, and of course the DNA has two strands woven together in a helical formation. And in order to make something from that, to decode those instructions, those two strands are unwound. It has an antisense strand and what's called a sense strand. Well, that antisense strand is read. It's read by this crazy good stuff called RNA polymerase, which is matching up letter for letter as it goes across this thing. Now, slight difference between DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. DNA, RNA. What's the diff? DNA consists of these four bases down here, T, G, A, C, A, and G. T, C, A, G, or T, G, A, C, doesn't matter which order. Those four right there, and if you saw the movie Gattaca, that was a reference to those letters right there. And so RNA, it only differs in that it has this thing down here as a uridine in case instead of the thymidine for the T. So that's it. RNA looks just like DNA except it has a U instead of a T. So when we read an mRNA strand down here, we see all of these letters here. U, A, C, U, G, C, C as we're reading that strand down there. Okay, those are the instructions. So when you get an mRNA vaccine, you get a piece of instruction that looks like this down here. And then that is injected into your body. It's a big, big, long string. It's like 3,800 letters long. It's a big piece of instructions. And those instructions are wrapped up. A whole bunch of them are packaged up in these little lipid nanoparticles. Together, those make a solution that gets injected into the deltoid muscle. And then the idea is that those packages are taken up by cells. Those instructions are read. Those instructions encode for, in what was the case of the vaccines, for the spike protein. Once that spike protein has been encoded and made, then your body says, wrong, let's fight that thing. Okay, is that background necessary? You bet. You know I'm going somewhere with this. So let's go there. This is a kind of a bland title, Immune Imprinting, Breadth of Variant Recognition, and Germinal Center Response. I'll tell you what Germinal Centers are in just a second. They're very cool. In human SARS-CoV-2 infection and vaccination. Okay, as mentioned, a lot of authors on here coming from places like Stanford and Ulaanbaatar in Mongolia. We've got people from Advanced Cell Diagnostics in Newark, and on and on and on. A lot of people, but kind of shocking findings that came out of this. I was not prepared for what happened in this paper. Again, I'll help you decode and understand the science speak, because scientists tend to speak in sort of bland terms whenever possible, and they couch things. So Punchline is in the title here. The mRNA from the vaccines is still found for up to 8 weeks in some patients. And so here in the summary of their paper, they read, during the SARS-CoV-2 pandemic, novel and traditional vaccine strategies have been deployed globally. Well, some of them very novel. We investigated whether antibodies stimulated by mRNA vaccination, so that's either the Pfizer, including third dose boosting, differ from those generated by infection or the other types of viruses, which are adenoviral, which would be the Oxford and the GAM COVID vac, or inactivated viral vaccines. Okay, so those are the three big things out there. You got your inactivated viral vaccines, you've got your attenuated virus vaccines, and that's the ChAdOx there, which is the Oxford, or you've got your mRNA platform. So those are the three big things. They looked at them all, really huge amount of work in this, well done, lots of controls. They showed the methods in this paper in ways that I could follow, even if I didn't understand all the cool things they're doing, because these labs are doing some amazing stuff. So, yeah. Oh, let me get this back here. So, very cool. Continuing, quote, we analyzed human lymph nodes after infection or mRNA vaccination. So, that's really cool. So, they got a lot of lymph nodes, and they took them out of people, including controls, and they were able to look at pieces of those lymph nodes for types of mRNA or proteins to say, what's in there? What's actually going on in here? And what differs between these things? And what can we tell? What they found, continuing, quote here, is viral variant infection elicits variant-specific antibodies. So viral variant infection elicits variant-specific antibodies. So if you had a specific viral variant, you would, guess what? You'd make antibodies for that. Not really surprising there. And they say, carrying on though, but prior mRNA vaccination imprints serological responses towards Wuhan Hu1. That's the original classic version of the virus back from January of 2020, rather than variant antigens. No surprise there, but now they've studied it and we have the data to go along with it. When you get the vaccine, you're getting a vaccine that imprints your body on a very specific type of the virus out there. No surprise. Now it's in a peer-reviewed journal called Cell, which is pretty awesome. However, the big part here that I want to talk to you about today is in red underlined here, continuing, quote, in contrast to disrupted germinal centers in lymph nodes during infection. Again, I will tell you what those are in just a second. Quote, mRNA vaccination stimulates robust germinal centers containing vaccine mRNA and spike antigen up to 8 weeks post-vaccination in some cases. So it's not all cases. Again, they couldn't do this on hundreds and hundreds of people, but in the cases they looked at, they found some of them, the mRNA was still there, fully detectable up to 8 weeks post-injection. As well, spike proteins are still out there 8 weeks post-injection. Not a surprise if you find the mRNA, pretty good chance that mRNA is being translated and it's being expressed. So that would give you the spike protein. So, but it means for some people, it's not like you get the shot and you get this little antigen sort of production response inside your body mounts a response to that, but these things go away. Now we know that for some people, these things are kicking around up to 8 weeks later. And that's as far out as the study went. Could it be 10 weeks? Possibly. We don't know. Hasn't been studied yet. So that's the finding. So why is this really important? Well, let's take a little detour here. What's a germinal center? What is that? Germinal centers, GCs, they are transiently formed structures within the B cell zone, or these are their follicles, in secondary lymphoid organs. So you have a lymph node, might be a big giant thing. Sometimes you can feel them when they get swollen, right? If you went into that lymph node, you would see things that look like this. These are the germinal centers here. Here's one and here's one. So you see all these big blobs here. So those are areas where the B cells come together and they organize for a really important function. I'll tell you, it's so cool. It's really cool. So we find these in secondary lymphoid organs. These are in lymph nodes, ileal, Peyer's patches, ileals, the ileum's down in your gut, the spleen, where mature B cells are activated, where they proliferate, differentiate, and mutate their antibody genes through somatic hypermutation aimed at achieving higher affinity. These are little training camps. These are the training camps when your body's saying, what do we have to fight out there? It turns out they hold this big organized rave party. They bring in all these different types of cells. They have this wildly complicated little dance they do with each other is they present antigen and they try it out and they're trying to make antibodies against it, and some work and some don't. Then they find the ones that work and they amplify those. It's just wildly complicated. It's beautiful. It's beautiful too. Here, let me show you maybe another picture of that. So here's a blow up of just one of these little balloons, the germinal centers here. And there's all these different cells in here, right? All these different types of cells. There's dendritic cells. There's follicle cells. We got these T cells. There's B cells. There's all kinds of different things. And there's different zones inside of this thing. There's a light zone, they call it, and a dark zone. You think you can see the light zone and the dark zone, right? So here, dark zone, light zone. Love how we named things back in the day. So at any rate, in these germinal centers, there's lots going on, but here's the primary thing. Oh yeah, I got a picture of this. Cool. So what happens is your body has some sort of a thing it wants to make an antigen or antigenic response or a full immune response to. B cells are part of that. B cells make the antibodies. That's all we hear about, right? Somebody gets an injection. What's their antibody titer? How high are the antibodies? Well, what happens is when you first get exposed to something, be it a spike protein or a piece of dirt that gets under your skin or a new bacterium, you have what are called naive B cells. So why are they naive? Because they don't know what's going on. These naive B cells don't have a clue. They don't know how to make an antibody against this thing that's invading. So the naive B cell comes and wanders on in here, and it goes through what's called clonal expansion. So it just starts dividing. It makes lots of clones of itself. This is the clone wars. So this naive B cell makes a lot of itself. So now there's a bunch of copies, like millions of copies of it. And then it goes through what's called somatic hypermutation. So they undergo self-imposed mutation. They scramble. They have these genes that are just designed to scramble and auto-scramble. And they go into this auto-scrambling function. And then the whole idea here is that some of these have improved affinity, higher, tighter binding. Their antibodies work. They work really well. Those are found. Those are selected for. Another set of cells comes along and says, you're a keeper. Come on over here, you higher affinity, wonderful B cell you, and let's expand you, and we'll make a bunch of you. And so they go off and they do that. This selection process here involves these dendritic cells. Some of them, they mutate, and guess what? That doesn't work out. They make disadvantageous mutations. They make antibodies that are kind of crap. So what happens to those? They undergo apoptosis, which means programmed automatic cell death. That cell says, it didn't work out for me. So it takes itself out of the game. Very polite of it. And then there's they go into a class switching, and then it all differentiates, and then you have these plasma cells that go out and they wander around in your blood, and they make lots of antibodies of different types against these invaders. And then they also differentiate down into these things called memory B cells, where they remember. We found out that people who'd been exposed to early SARS itself, SARS classic, the classic SARS, possibly also came from a lab now that we understand the data a little better. But at any rate, 17 years after that experience, they found that people still had a memory in the T cells and presumably in the B cells. They have memory. Your body retains the memory of those things. It's kind of like if you got a polio vaccine or if you got exposed to measles once or chickenpox once. Your body retains the memory of that experience in things like the memory B cells as well, memory T cells. So what am I trying? Why this little impromptu tour at a very sort of casual level through immunology? Because I want to impress on you. I really want to impress this on you. It's really, really complicated how the immune system works. This is why we would normally test vaccines for a very long time, because it's really wildly complicated, because a lot of things can go wrong, because we don't fully understand it yet, because it's pretty impressive how it operates. And so sometimes you get surprising things. Complex systems don't have predictable outcomes. They have emergent behaviors. So that's why we test these things. Well, we didn't have time. We did the warp speed. We pushed a lot of these vaccines out, and now we're finding out things. And this would be a thing that was just discovered, was that we find now that people, in some cases, have mRNA for up to 8 weeks after the injection and possibly longer, and they have spike protein 8 weeks, certainly after the injection, possibly longer. So let's go on back here. just complicated, and it's beautiful in its complexity. It's just beautiful, and we should have some humility around it. Now, here's why it's so beautiful. This is a 3D model of a spike protein from a coronavirus. You see the shape of that thing? An antibody works when it fits around that perfectly. Like, imagine like a lock only can be opened by a very, very specific key, because that key has a very specific set of ridges and bumps, or it might even have dimples and divots. It might even have magnets on it, depending on how complicated the key is. But that's how it works. That key has to fit perfectly into that lock, right? Anything that doesn't quite fit, and it's not, that key won't work. So kind of like that, what we're looking at here is called 3D topology, the surface, every little bump on this thing. An antibody has an exactly corresponding negative space to that positive space, and vice versa for all those little bumps out there. Now, if that was the only thing that the antibodies had to do, like, how would you do that? How would you design a machine that could figure out what the surface of something is, read it very carefully, and then design something that fits right on that? Well, you probably could if you had like computers and CAD machines and laser surface mapping, but these are just cells, and they kind of grope around and sort of feel these things, and I don't know, it's such a mystery. Anyway, so the antibodies fit perfectly onto just one little piece of this, right? So the antibodies might, I don't know, map onto like this little part right here, and the antibody would stick to there. Another set of another antibody might stick right here. If that was the binding site, that would be a neutralizing antibody. But again, wait, that's not all. It's also true that the surface of this thing doesn't have a 3D shape. It also has charges on there, plus positive and negative charges. And the antibody has to map onto that too. So it's kind of like, this is a very complicated lock and key scenario where the charges have to match, the shape has to match. It's very, very complicated. So that whole process of making those antibodies, that's what's going on in the germinal centers. All right. With all of that, now we can understand, I think, what this paper is telling us here. Results. What do they find here? Well, the mRNA is found in the lymph nodes. So in yellow, quote, the biodistribution, that's within your body, quantity and persistence of vaccine mRNA and spike antigen after vaccination and viral antigens after SARS-CoV-2 infection are incompletely understood, but are likely to be major determinants of immune responses. That's that science speak I was telling you about, like, that's careful sort of speechy stuff they did right there. Okay, but that's absolutely the case. Yes, it is incompletely understood, which is a fancy way of saying we're kind of figuring this out as we go. And thank you everybody who signed up and got these injections, because we didn't really know where these things were going to go in your body, or how many were going to be made, or how long they were going to hang around. Don't know any of that. And it's actually true. I talked about this many, many months ago, that there's going to be a bell curve. Some people are going to mount almost no response, right, and make almost nothing. Other people will have a fairly typical response, center of the bell curve, and then other people are going to have this really crazy response where they just make a lot of spike protein, or it really goes on walkabout in their body or whatever, and they're going to have a very different experience than somebody else who's on the other end of that bell curve. All right. Carrying on down here, they say we performed in situ hybridization. So that just means they took cells in tissue, and then they took pieces of mRNA and probes and antibodies and things that could find things in there, and they hybridized them in situ. So that would be for the RNA. They did this with controls, and they did it with SARS-CoV-2 vaccine mRNA specific probes. So that's cool. And they did the core needle biopsies of the ipsilateral axillary lymph nodes. That means if you got the shot in the left arm, they pulled the lymph nodes from the left armpit. That's fancy, ipsilateral axillary lymph nodes. That were collected 7 to 60 days after the second dose of either the Moderna or Pfizer vaccination, and, quote, here in yellow, detected vaccine mRNA in the germinal centers of the lymph nodes on days 7, 16, and 37 post-vaccination, with lower but still appreciable specific signal at day 60. Wow. Only rare foci of vaccine mRNA were seen outside of the germinal centers, so they were really the vaccine wandered right into those germinal centers, which is actually kind of probably where you want it. So that's a good thing. That's right where that's where it should show up. Hopefully, we wouldn't want to see it somewhere else, like I don't know, the brain or the heart or something, right? And I'm not sure they tested that. They were looking within the lymph nodes that they did get. They were asking, where did we see this stuff? And they mostly saw it in the germinal centers. I don't believe I read this pretty carefully, but I don't think they tested other tissues. I think there's another study for that. All right, and again, they did core needle biopsies of non-vaccinees, so 3, and COVID-19 patient specimens were also negative for the vaccine probe hybridization. So that means that they had 2 sets of controls, really. Controls who hadn't been infected or injected, and then people who had COVID-19. And in neither case did they see their vaccine probes stick to and light those up. Here's what it would look like. Here's what it looked like. You would see pictures like this. So this is a picture at many hundreds magnification, 600 magnification, they're saying there if you tip your head that way. And so here, this is a picture from within a germinal center, and we're looking at 2 things. First, if you can see, and I hope you can, let me know if you can't, but anyway, the link to the papers is always down here, so if you want to go and see these images in high resolution, just click the link. Link's also down in the description below. If you can see, there's a lot of blue things in here and some really dark blue blobs, but in between them, there's sort of these tannish, greenish little signatures you can see in there. I won't, maybe you can just stare at that and see, but that's the spike protein. So the spike protein has been stained, and it stains it shows up as sort of a greenish, brownish sort of a smear there, but there's plenty of it, and you can really clearly see it here. And then the mRNA vaccine probe lights up with a much darker signal, easier to read all these purple dots right here. And you can see these dots at a slightly lower magnification like this. And this is what it looks like when you look across time. So here's their control. So they're just doing a standard mRNA control probe, and these are the germinal centers, and you can just see this stuff lighting up, lights up, lights up. So they're the way they do their in situ hybridization works. That's good. And then when they did it with their mRNA vaccine probe, you see it here on day 7. You see all these dots in here at day 16. You really see it very strongly at day 37. You can still see a few dots out here at 42 and even at 60, but not a lot. So it is being degraded away. And then as well, we don't really see a lot of spike protein here at day 7. We do see quite a bit at day 16. I don't see a lot in the day 37 image, but it's there. There's some here, and there's also some, actually quite a bit out here at day 60. So let's just zoom in a tiny bit on that last one there so you can see it. Out here at day 60, we do see a lot of those greenish, tannish, blobby things out there. So spike protein, 60 days post-injection, still out there. Why is that a concern, potentially? I mean, it's a good thing if you want to make a lot of antibodies. It's a concern if that spike protein itself is toxic. There's concerns that it is toxic. There's a lot of data suggest it is toxic. So how much you would make, those are the 3 things you would really want to know in a program which really wasn't done in warp speed, which is when we inject the same amount into people of all different sizes. Everybody gets 30 micrograms of Pfizer, then another 30 micrograms, grand total of 60 micrograms. Didn't matter if you weighed 85 pounds or you weighed 495 pounds. You got 30 micrograms and 30 micrograms. That's part 1. Part 2, we'd want to know, did it stay where we thought it was going to stay? Did it go on walkabout? Was there anything we should have known about maybe aspirating the needle backwards to make sure that we weren't in a vein so that this stuff didn't just sort of like go somewhere it shouldn't have all through the body? Things like that. And then thirdly, you'd want to know, well, how much stuff did it make? How much spike protein was actually made? Because that's the thing we're shooting for in this particular adventure. And then the final thing you'd want to know is, well, how long does that spike protein last? Because if you could find spike protein a year later or 2 years later, you might say, ooh, that's too long. Like what you really want is a recurve of a response. It's not what we're seeing. We're seeing 60 days later, there's still a few dots out here. Here's a dot way up here. So there's still some mRNA, and there's quite a bit of spike protein 60 days later. Now, why does that matter? Well, as a reminder, we were assured over and over again that the vaccine mRNA was very short-lived in the body. We saw things like this, where if you went to the Nebraska Medicine channel here on July 2nd of 2021, They ask and answer the question, how long mRNA lasts in the body? And they say the Pfizer and Moderna vaccines work by introducing mRNA into your muscle cells. Not sure if it's into the muscle cells. Must be around them. So I don't think that's quite a good sentence. Second, in yellow, quote, the cells make copies of the spike protein and the mRNA is quickly degraded within a few days. The cell breaks up the mRNA into small, harmless pieces. mRNA is very fragile. That's one reason mRNA vaccines must be so carefully preserved at very low temperatures. And then they say the spike proteins that were generated last up to a few weeks. So that wasn't a bad guess early on. Now that we have the data, we know it's up to 8 weeks. Here's one, a Q&A from UC San Diego. Are COVID-19 vaccines safe and effective? And Dr. Robert Chip Schooley is answering the question. So they're really looking here, are they safe? Are they effective? How do they work? Are there any potential side effects? What should I do? So Dr. Chip Schooley is, let's see what he said. He's confidently wrong. How could that be? He wrote, once these mRNA instructions are delivered, the mRNA is degraded over 12 hours. And the spike protein that is created as a preview for the immune system disappears over 48 hours. I'm not sure how Chip was that confident to say things like that back then. I would have personally softened that up quite a bit. He might say, I've read, Pfizer told me, Moderna says that the mRNA is degraded over 12 hours. I would suspect that it would be degraded quickly because mRNA tends to degrade quickly. But the truth is, if Chip had looked into this more carefully, he would have understood that this isn't actually typical mRNA. Remember, I told you, it's got that U as part of the RNA nucleotide sequence, right? And so that UCGA, the U in that is uridine. They use something different when they made these. They use something called pseudouridine. It makes a very much more stable mRNA product, and apparently very stable, which can last in the body up to 8 weeks. I'm surprised by that. But again, we read a lot of confident declarations like this that said, oh yeah, this stuff's just going to be gone in a matter of hours. I saw lots of that. As well, Dr. Schooley here also, I think, confused absolute risk with relative risk, where he said Moderna and Pfizer vaccines are 95% effective against the original variants and at least 90% effective against the new variants. That, of course, is a relative risk reduction, not an absolute risk reduction. In fact, also didn't seem to be aware of the fact that these things degrade and wane in effectiveness. That's okay. It was July. We were still learning some things out, but now we know a lot more than that, so I expect to see that corrected. At any rate, here as recently as September 2nd, it says here, you don't need to worry about COVID vaccines. The mRNA vaccine, the mRNA gets degraded relatively quickly, and after a few days, there is unlikely to be any mRNA vaccine left in our bodies. That's the Hindu business. So they at least couched it with the word unlikely. Other places didn't do that. So the ec.europa.eu said, the vaccine mRNA will not enter the nucleus of the cells where our DNA is. Once injected, the mRNA enters a human cell. It degrades quickly. Only stays in the body for a matter of days, they said here. And down here on the Ministry of Health in Singapore is this next one, general information in COVID-19 from their, still says this today. This is pulled from the January 17th is the last time they updated this, of 2022. They say here that the mRNA is completely degraded within 48 hours of introduction to the body. So they're going to have to amend that, of course. We'll see how quickly that gets amended. And then if we go to Wikipedia, which is, I hold in kind of low opinion now because they've somehow confused science and politics, and in a regrettable way. At any rate, they write in Wikipedia, which some people consider authoritative, quote, the body degrades the mRNA fragments within a few days of introduction. So that'll obviously need updating as well. All right. Why did I bring that up? Because at the time, back when the vaccines were first coming out, I made a point of saying we need to know how long does the mRNA stay in the body and how long will the spike proteins kick around and how much of them are made in the body. And I got pretty sort of censored and dismissed from that. I was told that these were, baseless speculations. I'm like, doesn't seem baseless to me. They're like honest questions. That's how science works. If you don't know something, you shouldn't be confidently incorrect about it. You should couch things. And at any rate, of course, all scientists agree when you censor the ones who don't. Hey, I agree with that. Here are my conclusions for episode 2047. The mRNA in the Pfizer and Moderna vaccines persists in some people for at least 8 weeks post-injection. The resulting spike proteins also persist in some people for 8 weeks post-injection. Both the mRNA and the spike proteins do not stay at the injection site, but they travel to nearby lymph nodes. That we know at least that much. We don't know where else they might go. We need more studies. That's science. We were told consistently and confidently by people like Chip that this wouldn't be the case. We might question, how are these people so confident? And why were they so confident? It's kind of odd. And this, of course, confirms that all mRNA vaccinated people are actually test subjects in a large real-world experiment. And it really shouldn't have been this way. We should have easily asked for and demanded and gotten a full set of studies. This is why vaccine development takes usually 10 years. You would do studies like this either in a limited select set of volunteers, but this is after you've already done complete tests on mice and rats, and then you might work your way up and do ferrets, go a little bit higher. You might actually do primates, and then you would do humans, and you'd be studying, where does this stuff go? How long does it last? Are there any side effects we don't know about? What happens after 2 years, 3 years, 4 years post-injection? What happens after we give a 6th or 7th or 8th booster? What happens to reproductive? Is there anything we could notice neurologically? Is there anything developmentally in the offspring? Those sorts of studies all take time. There's just no way you can throw money at that and make it shorter. That would be like saying, well, if it takes one woman 9 months to have a baby, it should take 9 women 1 month. It's not how things work. So we short-circuited the whole process, but guess what? Nobody's doing those studies today because we're running real-world experiments out here in the world of humans, and we're finding things out. For the most part, I think the vaccines are fine. But for a few people, they're not. And I think this data begins to explain why that's the case. These few people may be people who are making just constant amounts of spike protein. And we need to know, is there a way to turn that machinery off? Is there something we can do? Is there a way to minimize whatever the toxic impact they're experiencing from that? But here's the summary. The immune system is wildly, wildly complicated. And it's much as we know about it, we probably don't know 10 times that amount about it. We're going to learn more. That's fine. But we ought to have the humility that says, we don't really know about this. It's really actually quite complicated. And to pretend as if it's as simple as saying, are you pro-vax or anti-vax is really ignorant. And the correct way to look at this is to say, look, we only know so much about these things. We can say on balance that there's some positive benefits used in certain ways in certain applications at certain times. I think we can now say that putting a leaky, non-sterilizing vaccine out is a broad matter of public policy, up to and including mandates that were forcible in their construct, was not the right thing to do. We really should have had a targeted approach. We should have been giving these things very preferentially to people where the risk profiles clearly indicated that. Age, comorbidities. Those were things that were quite easily determined, and there are countries that are running those sorts of policies right now. But for people who are clearly not at risk from COVID, below a certain age, right? Call it 30. Below a certain BMI, call it 30, right? If you were below the double 30s in this story, you really probably ought not to be getting the vaccines at this point from a risk standpoint. If the vaccines were sterilizing, if they prevented transmission, if, and a bucket of roses, right? Then I would say very different things. But at this point, where we are in the case of Omicron, where we are in the absolute, not only non-sterilizing way that these vaccines are interacting with Omicron, but seemingly double jab people actually catch Omicron at a faster rate than non-jab people. Given what we're seeing in terms of the overall response, guess what? We're finding things out about these particular vaccines all the time. So those experiments are being run. By the way, I have a super important part 2 to this that I'm going to be talking about. Hey, if you like this, you know who I am. I'm going to do my level best to teach you something. I'm going to do my level best to get what's in here out in a way that you can acceptably have it land in your head in a way that you can take it off with you. That's what I do. I'm a natural teacher. It's how I'm a communicator. So I spend a lot of time trying to figure out what's going on in the world. This concept here, the common knowledge game, which comes to us courtesy of Ben Hunt, is phenomenal. It's a great piece of framing that helps us understand the world, and it helps us understand where we're going. By the way, things are a little cray-cray out there in case you hadn't noticed. I'm sure you have. So everything that's going on in New Zealand, Australia, Canada, United States, across big parts of Europe, but not all Europe. Some of them have caught on and decided to move on from this whole COVID fiasco. But understanding where we're going, this common knowledge game, is actually really critically important. So you'll find that over at Peak Prosperity, and I will be back there with everybody talking about that idea. It's a good one. It's a lot of fun. So that's all I have for you today. Thanks so much for listening. It's a lot of fun sharing this science with you. I hope you got something from it. Now you know what a germinal center is. Maybe you didn't want to know, but now you do. All right. Hey, thanks for being here, everybody. We will see you next time. Bye.